Genetically modified salmon is actually quite weird to find in the market. In fact, Actividad has started producing genetically modified salmon 2 years ago. However, people are still skeptical about whether to buy it or not. Likewise, a big part of this problem came up due to some pandemic delays and organizations that were totally against the production of such a product like this. It was mainly intended to sell these products in supermarket chains such as Walmart, Costco, etc. Although the FDA has approved the production of sterile genetically modified salmon to avoid any substantial risk to the environment and of course to the endemic species of salmon, there are still some concerns on the probable risk this product may represent for human health. On the one hand, farmed salmon usually contains higher amounts of less healthy omega-6s fatty acids and acceptable rates of omega-3s fatty acids, which is a healthy one. On the other hand, genetically modified salmon produced by Actividad farms do have much higher rates of the healthy omega-3s fatty acids than conventional salmon, of course.
The developed genetically modified Atlantic salmon has a DNA construct containing the promoter and terminator regions of the antifreeze protein gene from the ocean pod linked to the growth hormone coding region, complementary DNA clone from the Chinook salmon. After microinjection into fertilized, non-activated Atlantic salmon eggs via the micropyle, transgenic Atlantic salmon were generated. The presence of the transgene was detected by Xiaojun Du and colleagues in 1982 using PCR with specific primers. Thus, first, it is a gene construct entirely derived from fish DNA sequences. Secondly, the transgene allows expression of the new growth hormone gene in the cold autumn and winter months when the antifreeze protein gene is naturally activated. Thanks to this, Actividad salmon manages to maintain growth throughout the year. The result of this whole process is that Actividad salmon grow much faster than natural salmon and therefore reach the same commercial size in about 18 months, half of the ones not modified. For safety, only females are produced. Moreover, all of them are produced as triploid organisms and therefore all of them are sterile. The growth of transgenic salmon eggs takes place away from the coast and any river or lake, again for additional and redundant safety measures. In the unlikely event of an accidental release or escape, the field experiments carried out show that these salmon, due to their constant growth, need a constant feeding, difficult to find in a natural environment. Therefore, this Actividad salmon, besides not being able to reproduce or interbreed with the rest of the salmon, would not adapt to the natural environment and would quickly be displaced by natural salmon. Therefore, the FDA's conclusion was that the production of Actividad salmon does not have a significant impact on the environment.
Another genetically modified product is corn. Bt corn is an insect-resistant crop that carries genes from the soil bacterium Bacillus thuringiensis. Its genome contains a gene that produces a protein called the Bt delta endotoxin. This protein is formed during sporulation in Bt strains and aggregates to form crystals. Such cryotoxins are toxic to specific species of insects. At first, Bt was sprayed on fields as a pesticide. However, it got washed off by rains, and the Bt toxins broke down quickly when exposed to UV light. So, to address this problem, genetic engineering has been used to incorporate delta toxins into corn. Bt corn is an alternative to spraying chemical insecticides because it offers environmental and economic benefits to farmers. Actually, according to FDA estimates, more than 92% of U.S. corn crops are planted to this technology.
As mentioned, to create a Bt corn, a gene from a cry-IC for a particular Bt toxin is selected and inserted into the cells of a corn at the embryo stage. It must be inserted with additional genetic material, including a promoter sequence that will determine how the gene will be expressed in the plant. This results in a mature plant that has the Bt gene in all its cells and expresses an insecticidal protein in its leaves. There is a marker gene that allows plant breeders to easily determine which plants can be transformed. Herbicide and antibiotic tolerance promoters are commonly used to identify transformed plants. There may also be a plasmid or vector sequence that allows rapid multiplication of the gene of interest in a bacterial host prior insertion of the crop plant.